Hi, this is Roy Henson. I'm going over lesson 5 and going over pretty much how this lesson works. And we're just going to go over the inheritance of linked genes, and we're going to understand what is meant by genetic linkage, examine scientific evidence for genetic linked genes, understand why test crosses are used for genetic linkage to determine gene order and map distances. It's a practice problem that I figured out using tri-hybrid crossroads, and understand how linkage can be used to create genetic maps. So I just got to get my glasses here so I can see. Let's begin. So first, we're going to go through evidence-based linkage for Bateson and Pruitt. Evidence-based, and pretty much it just goes through that the red flowers and purple flowers are linked because red is recessive, homozygous recessive, and purple is homozygous dominant, and they make purple flowers, which is here. But they actually make a certain number of observed. So they did quite a few of these to make sure the statistics are right. And they figured out that there was this with round pollen and long pollen, and they did this, and they found all these different this ratio observed, the 15 to 1 to 4, 1 to 1.5 to 4.5. And what they found is that there's something going on here. Examine inheritance of flower cover, and they noticed that these two are linked, and they're close together, or they're related somehow. The round pollen and the purple flower, or the flower color and the pollen, they're related somehow. And they found out they're linked. The genes are so close together that they affect one another on the chromosome. And so the linked genes, as it says here, violates the law of independent assortment, and they're transmitted to offspring as a single unit. So the genes are so close together that they transmit together. It's on the same pair of homologous chromosomes. And so this linking and this crossover, as a review of a previous lesson, it's during prophase 1. There's synapses, and these homologous chromosomes line up right here, and this is the synapse. This is lining up here, and then they have a synaptical cortex, a protein that holds homologous chromosomes together, and then they cross over, and it occurs at a chresmata. And so that's what this is here. They're crossing over, and that's kind of a review from earlier. And as you can see from crossing over, you can end up with all these different types of chromosomes. But as you can see, they're linked. They're linked together, so close together that they cross together. And so in the presence of crossing over, a new linkage pattern is established, non-parental or recombinant, and the original pattern is maintained, parental or non-recombinant. So you either end up with the same genes as the parent, or you have some crossing over that happens. And so test crosses, and so these the frequency of recombination is related to the physical distance between the gene gleans, and we should be able to calculate this distance between the linked genes by analyzing the frequency of recombination between the genes. It's talking about how many offspring and the ratios. And then it goes test crosses are used to determine the distance between linked genes. Crossing over the homologous parent does not change the arrangement of the alleles. We can also determine the distance between the linked genes by analyzing recombination heterologous parent. And so it's saying that we can have these genes linked, and we can look at those statistically. And then we can also determine the map distance. We can determine the distance between the linked genes. Actually, if the linked genes are more than 50, then that's considered a non-linked gene. But if anything less than 50, they're considered linked. And then it just goes to the map distance, the number of recombinant offspring, the total number of offspring, and it'll give you in map units. And I'm actually just going to skip this one and go right on to the example in linkage, and I'm going to explain this real quick for you. And so we have 3 different traits here. We have thread is dominant to wild type is dominant to Rosie, and wild type is dominant to Harry. It really doesn't matter. We just need to, from what I understand, we just need to know the difference between the two. And so some are heterozygous for all 3 genes, some are not. And so what I did is I actually took the slide and I ordered these in the most prevalent versus the least prevalent. And the most prevalent is actually the allele arrangement of the heterozygous. That's the answer to the first one. It says, what is the allele arrangement of the heterozygous parent? And how you do that is you just take, I marked wild type with pluses, and I write non with non, and so you just take the highest one, so 311. So that would be Harry, which is not wild type, or Harry, which is wild type, or Harry, and then it's Rosie, and then wild type there. And so, sorry about the confusion there. So I just take the most dominant one and I put it on the bottom, and then I take the next abundant one and I put it on the top. So this one is thread, wild type, and so that's how you determine the allele arrangement of the heterozygous parent is through that. This is it. That's the answer to A. And then it's asking us what is the gene order. And so how you do this is kind of interesting. Is you take the parent's one and then you take the middle, whatever one you decided to be the middle. You take the middle one. So I decided it to be R, and I take the R and I move it forward and then I move it back. And so as you can see, it's just the parent, and the R is moved to the first position and the R is moved to the last position. And pretty much what we're doing is we're determining the gene order. And what you do is, then what you do, you go to the least abundant one, and then you make a zigzag. So we know the least abundant one here is Rosie, wild type, and so we're just going to here and we're going to look for a zigzag where it's Rosie, Rosie and wild type. This one doesn't have it. Rosie, wild type, almost no. Rosie, wild type, and Rosie, wild type. I thought I fixed. No, this isn't wild type. This is Rosie, this is Rosie, thread, and Harry. This is Rosie, thread, and wild type, and this is Rosie, wild type, H plus, wild type, T plus. And so now this is the gene order, and you can also check it with the 9 one, because the 9 one should be this is be threaded, hairy, and wild type, which it is, thready, hairy, wild type. And so you know that this is your gene order right here. From your gene order, then what you're going to do is you're going to calculate distance between the linked genes. How you do this is you take your different types of gene here, and so we have this gene, which is 97. So wild type for all 3 genes, so wild type for all 3 genes, 97 plus the next one that's closest, the closest to it, so 97 to 83, and so 83 right here. And then you add it to the, and so what you're doing is you're determining the distance of between the middle gene, which is 9 plus 4. So you just take 97 plus 83 plus 9 plus 4 divided by 1,000 times 100, and then this will give you your distance between should be your threaded between your hairy, and then you do it for the other one too, and so you end up with the other ones here, 57 and 47 here and here, and then 9 plus 4, and you end up with 11.5. And so these are your distance between your linked genes. You have H in the middle, and you have 19.3 on one side and 11.5 on the other. And so that's the distances between your linked genes. Then what's going to ask you, what are the distance, calculate the interference value. Now the interference value, it goes to this in the notes from what I understand, but I'm just going to run through it real quick. You take the distances between your linked genes, you take them over 100 and multiply them by themselves, and then you multiply it by the total number of offspring. So I made a calculation here, total number of offspring, and then this will give you a pretty much a percentage, or a number. And then you take this number and you take it and you add the least 2 together, which is 13, 9 plus 4 is 13, and you divide it by the number you just found, and you take it by -1 and times by 100. So just a little formula right here. So you get this number came from here, 13 came plus 9 plus 4 times 100 is equal to 41.4%, and that is your interference value. And that's how it's done. That's how I do it. That's just the run through I do it. But yeah, I get them right. So then just going to continue on with the and then it goes to the steps. You determining the gene order map distance between linked genes. You set up a trihybrid test cross. You determine the allele arrangement of the heterozygous parents. So you set up a trihybrid test cross. You get all your statistical value of all your different offspring. You determine the allele arrangement of the heterozygous parent, and then you determine the gene order by the heterozygous parent, and you rewrite the test cross so that it's that you determine the map distance between one gene in the middle, determine the map distance between the middle gene of the third gene. You generate a linkage map. You calculate the interference, just like just like we did. Heterozygous parent, gene order, map distances, interference value. Once you do a lot of them, it gets pretty simple, and it's a lot easier. And so the theory regarding the chromosome mapping, the map distance is in MUs and MCs. So this is map units, and I don't know what this one is an estimate of the actual distance between base pairs between the genes. As the actual distance between the genes and base pairs increases, the map distance, or MUs, becomes increasingly inaccurate. Like I said, once you get over 50 MUs, they're not linked. Double crossovers and distance restore original gene arrangement. If the map distance between linked is more than 50, then they sort independently, just like I was saying before. And so this is a little show here showing that you can have multiple genes within a 50 base pairs apart, and you can get that. And so as you can see, there's this is a genetic map made in order distance between genes. It allows us to understand the organism of the genome, isolate a specific gene and study it, understand the evolution between organisms, identify diagnose genetic diseases, provide information from breeding experiments, frequency of non-parent and recombinant offspring is used to determine order and distance. So this is what we did in the previous problem. This is the fruit fly genetic map, and it looks like we've mapped out all the genes on a fruit fly. So this is actually the last slide. I thought this is a pretty easy set of slides. I hope that example helps you guys out with doing the problems. And this genetic map, I just think it's interesting. You can learn a lot. Thank you so much for watching, and have a great afternoon, day.